So you know CRISPR, right? CRISPR is this genome guidance and cutting mechanism. It's what George Church likes to call genome vandalism. So you basically are able to...
Good line.
You can basically take a guide RNA that you put into the CRISPR system, and the CRISPR system will basically use this guide RNA, scan the genome, find wherever there's a match, and then cut the genome. So, I digress, but it's a bacterial immune defense system. So basically, bacteria are constantly attacked by viruses, but sometimes they win against the viruses, and they chop up these viruses and remember as a trophy inside their genome, they have these loci, these CRISPR loci, that basically stands for clustered repeats, interspersed, etc. So basically, it's an interspersed repeats structure where basically you have a set of repetitive regions, and then interspersed were these variable segments that were basically matching viruses. So when this was first discovered, it was basically hypothesized that this is probably a bacterial immune system that remembers the trophies of the viruses that it managed to kill. And then the bacteria pass on, they sort of do lateral transfer of DNA, and they pass on these memories so that the next bacterium says, ooh, you killed that guy. When that guy shows up again, I will recognize him. And the CRISPR system was basically evolved as a bacterial adaptive immune response to sense foreigners that should not belong and to just go and cut their genome. So it's an RNA-guided RNA cutting enzyme or an RNA-guided DNA cutting enzyme. So there's different systems. Some of them cut DNA, some of them cut RNA, but all of them remember this sort of viral attack. So what we have done now as a field is, through the work of, Jennifer Doudna, Emmanuel Carpentier, Fang Zhang, and many others, is co-opted that system of bacterial immune defense as a way to cut genomes. You basically have this guiding system that allows you to use an RNA guide to bring enzymes to cut DNA at a particular locus.
That's so fascinating. So this is like already a natural mechanism, a natural tool for cutting that was useful in this particular context.
Yeah.
And we're like, well, we can use that thing to actually, it's a nice tool that's already in the body.
Yeah. Yeah.
And
it's not in our body, it's in the bacterial body. It was discovered by the yogurt industry. They were trying to make better yogurts, and they were trying to make their bacteria in their yogurt cultures more resilient to viruses. And they were studying bacteria, and they found that, wow, this CRISPR system is awesome. It allows you to defend against that. And then it was co-opted in mammalian systems that don't use anything like that as a targeting way to basically bring these DNA cutting enzymes to any locus in the genome. Why would you want to cut DNA to do anything? The reason is that our DNA has a DNA repair mechanism, where if a region of the genome gets randomly cut, you will basically scan the genome for anything that matches and sort of use it by homology. So the reason why we're deployed is because we now have a spare copy. As soon as my mom's copy is deactivated, I can use my dad's copy. And somewhere else, if my dad's copy is deactivated, I can use my mom's copy to repair it. So this is called homologous-based repair.
So all you have to do is the cutting, and
That's
you don't have
exactly
to do
right.
the fixing.
That's exactly right. You don't have to do the fixing.
Because it's already built in.
That's exactly right. But the fixing can be co-opted by throwing in a bunch of homologous segments that instead of having your dad's version, have whatever other version you'd like to use.
So you then control the fixing by throwing in a bunch of other stuff.
That's exactly right. And that's how you do genome editing.
So that's what CRISPR is.
That's what CRISPR is.
When in popular culture people use the term. Wow, that's brilliant.
Yeah.
That's
so CRISPR
awesome explanation.
Genome vandalism followed by a bunch of Band-Aids that have the sequence that you'd like.
And you can control the choices of Band-Aids.
Correct.
Yeah.
And of course, there's new generations of CRISPR. There's something that's called prime editing that was sort of very, very much in the press recently, that basically instead of sort of making a double-stranded break, which again is genome vandalism, you basically make a single-stranded break. You basically just nick one of the two strands, enabling you to sort of peel off without sort of completely breaking it up, and then repair it locally using a guide that is coupled to your initial RNA that took you to that location.
Dumb question, but is CRISPR as awesome and cool as it sounds? I mean, technically speaking, in terms of like as a tool for manipulating our genetics in the positive meaning of the word manipulating, or is there downsides, drawbacks in this whole context of therapeutics that we're talking about, or understanding and so on?
So when I teach my students about CRISPR, I show them articles with the headline, "Genome Editing Tool Revolutionizes Biology." And then I show them the dates of these articles, and they're 2004, like 5 years before CRISPR was invented. And the reason is that they're not talking about CRISPR. They're talking about zinc finger enzymes that are another way to bring these cutters to the genome. It's a very difficult way of sort of designing the right set of zinc finger proteins, the right set of amino acids that will now target a particular long stretch of DNA. Because you know, for every location that you want to target, you need to design a particular regulator, a particular protein that will match that region well. There's another technology called TALENs, which are basically, just a different way of using proteins to sort of, guide these cutters to a particular location of the genome. These require a massive team of engineers, of biological engineers, to basically design a set of amino acids that will target a particular sequence in your genome. The reason why CRISPR is amazingly, awesomely revolutionary is because instead of having this team of engineers design a new set of proteins for every locus that you want to target, you just type it in your computer and you just synthesize an RNA guide. The beauty of CRISPR is not the cutting, it's not the fixing. All of that was there before. It's the guiding. And the only thing that changes is that it makes the guiding easier by sort of, just typing in the RNA sequence, which then allows the system to sort of scan the DNA to find that.
So the coding, the engineering of the cutter is easier on the in terms of SP. That's kind of similar to the story of deep learning versus old school machine learning. Some of the challenging parts are automated. Okay, so but CRISPR is just one cutting technology, and then that's part of the challenges and exciting opportunities of the field is to design different cutting technologies.
Yeah.